lecture of the year. I'd like to celebrate and acknowledge the traditional owners and custodians of the land on which we meet today. I'm coming to you from the lands of the Wurundjeri people of the Kulin Nation, and I'd like to pay my respects to their elders past and present, and to all Aboriginal and Torres Strait Islander people who may be with us today. So I'm Lizzie, and I'm a PhD student with Gemma and Andreas, and I'm going to be your chair for today's seminar. Our speaker today is Sir David Lane. He is currently a professor at the Karolinska Institute in Stockholm and chairman of the Chungai Pharma Body and chairman of the scientific committee of the Cancer Research UK and NCI Cancer Grand Challenge. So today he is coming to us from the UK, so we thank him for staying up so late to deliver this seminar on tumour suppressors. He is highly qualified in this area as he was one of the researchers that discovered the tumour suppressor gene P53. And you may have heard that the P53 being called guardian of the genome, and this is a phrase which comes from one of David's many reviews. He has played a huge part in UK cancer research through running his lab and his roles as Chief Scientist of Cancer Research UK and Scientific Director of the Ludwig Institute. He has won numerous awards over the years, and for his contribution to cancer research, he was knighted in 2000. His research and career has tied basic research with translational research. So during his time at A*STAR in Singapore, he was chairman and then executive director of the Institute of Molecular Cell Biology, where he added on translational focus to their basic research before becoming the chief scientist of A*STAR. He was also founding CEO of A*STAR's Experimental Therapeutics Centre. So we're really pleased to have David give us this seminar today. And so for the students, to ask a question during question time, we encourage you to use your raise the hand button in Teams, and I will call on you, but you can also type in questions into the chat box. If you do ask a question, Gemma will add your name to go into the list to go into this draw for the prize, and she has chocolates in her office on 5 West if any of you who ask a question want to come grab one. So David, thank you for coming today, and over to you.
Well, thanks very much, and it's very nice to be with you all. I'm a great fan of the WEHI, and I have often visited Australia for the lawn cancer conferences. It's a great shame I can't actually give this lecture in person, but it's still very nice to talk to you about it. It's interesting, I think, whenever you get a lecture ready, you start to learn a lot of new things yourself and look at things you thought you knew. So it's been fun getting ready for this. So I'm going to start with a bit of historical perspective. So this is a picture of Henry Harris, who was at the William Dunn School in Oxford. And he became very interested in using cell fusion as a way to study cell biology. And you know, one of the origins of the concept of tumor suppression came from this experiment that Harris and his colleagues carried out. And what they did was to fuse tumor cells and normal cells using viruses actually to promote the fusion, and then studied the behavior of the hybrid cell. And they found that these hybrid cells were generally non-tumorigenic, suggesting that normal cells contained information that could suppress their cancer cell phenotype, leading to the concept of tumor suppression or tumor suppressor genes. And actually, these are very interesting experiments. They've kind of become revived in a way as people have started to realize that actually these kinds of cell fusion events do actually happen in human cancer, and there's evidence of cancer cells fusing to normal cells in development of neoplasia. Though Harris reported the basic finding that generally the tumor cell was able to suppress the phenotype of the normal cell, that's not universal. And of course, we know a very important exception, and that is the fusion of normal B cells with myeloma cells, where the hybrid cell is actually able to grow as a cancer cell and continue to produce antibodies. That's the basis of hybridomas. But in any case, this was one of the first sort of concepts that led to the idea that there could be gene expression or protein expression that would suppress the development of the cancer phenotype. The other way that people began to appreciate the concept of tumor suppression was through the discovery of inherited cancer family syndromes. And there were a number of pioneers in this area, and one of these was Henry Lynch, and he's now known because his syndrome is named after him, Lynch syndrome, which is a form of inherited colorectal cancer. And he's a very interesting guy, actually. He worked in more remote parts of the United States, in Omaha and places like that, and he began to do his work because he met patients who were very pessimistic about their health and couldn't be bothered to sort of correct their behavior because they said, well, I'm going to die of cancer. Everybody in my family dies of cancer. And he began to investigate these anecdotal cases and began to assemble pedigrees of families which did indeed have very high incidence of cancer of particular types. And basically what he discovered was families in which there was a very early onset of cancer. And he was an incredibly hardworking individual, and over the years he collected over 3,000 pedigrees of cancers showing inherited susceptibility. I read about him a bit, for this lecture. A fascinating man and fascinating story. He had great trouble getting any grants. At the time, there was an absolute conviction all cancer was caused by environmental carcinogens, and the idea that it could be an inherited component was kind of very unpopular. So he had great struggles to get his work supported. But he persisted and became very, very well recognized and lived to, I think, 91, very grand age. What he discovered was families which showed a classic Mendelian inheritance of cancer susceptibility, showing the behavior of the segregation of a dominant autosomal gene. So in this case, all the individuals in blue would have developed colon cancer at a young age. And as we'll see, it subsequently emerged that one of the genes that he identified, or one of the syndromes that he identified, was caused by mutations in a gene that's involved in mismatch repair. So this was an example of a tumor suppressor gene that's controlling the fidelity of DNA replication. The other sort of very notable pioneer in this field, who also lived to a great age, is Alfred Nutson, and he studied retinoblastoma and discovered really very important things about the genetics of inherited retinoblastoma, and distinguished it from spontaneous forms of this type of eye tumor. This is an amazing paper. So he published a single author paper in 1971 in PNAS entitled Mutation and Cancer, Statistical Study of Retinoblastoma. And this was based entirely on clinical observation and interpretation of what those clinical observations meant. So he observed 48 cases of retinoblastoma that occurred in the hospital he was working at between 1944 and 1969. And what he analyzed was the age of onset of these retinal cancers, whether they were unilateral, that is in one eye, or bilateral in both eyes, and whether there was any family history of developing this type of retinal tumor. And he distinguished from that analysis that there were really two types of occurrence of this retinal cancer. The first type were inherited cancers, where there was a clear familial predisposition. And these tumors were nearly always bilateral, and there were often multiple tumors in each eye, and they occurred at a very young age. And in a given family, there would be many affected individuals. And then the second group of cancers that he discovered, the second sort of retinoblastoma, were ones in which the tumors were always unilateral, that is, only one eye was affected, usually a single tumor. These occurred later and were very rare in the population as a whole. So this core data he then analyzed, and this is a pretty amazing paper because the results are very small, but the discussion's very long. And what he deduced actually was quite extraordinary. He deduced two things. One, he deduced the mutation rate that must be occurring in these retinal cells. And the second, and most important, was the idea that he came up with, which is called the Nutson's two-hit hypothesis. So essentially, just by looking very carefully at this clinical data and applying statistical methods, he recognized that retinoblastoma is a very unusual tumor. There are very few mutations required for the tumor to develop. And this is a typical childhood cancer. In fact, if this cancer doesn't occur by a certain age, it will never occur. So it's a very clear, susceptible population of cells, these dividing cells in the retina that subsequently no longer divide. And so the risk of cancer is very specific to a particular type of tissue and particular type of cell, and also to particular periods in development. And what he realized is that the two forms of cancer could be understood in the following way. So there's a hereditary form in which what's inherited is a mutation in the gene responsible for susceptibility to retinoblastoma, so that all of these individuals are heterozygous. They have one wild type allele and one mutant allele, and therefore to enter the state in which they have no functional gene resisting retinoblastoma, they need to only have a single somatic mutation in the second chromosome. Whereas the non-hereditary, the very rare form, requires two mutations to occur, one in each parental copy of the RB gene or retinoblastoma gene. So, I mean, this was done entirely on the basis of genetic analysis. He didn't know what the gene was. He didn't know its chromosomal location. But of course, eventually the gene responsible for this defect was discovered, and a number of groups were able to show that exactly what he predicted was occurring. In other words, mutations were present in individuals with inherited susceptibility as a germline mutation. And then as the tumor developed, they lost the function of the second allele, whereas in the non-hereditary cases, there were two genetic events required because both alleles needed to be mutated in order to see the phenotype. So since those times, a number of tumor suppressor genes have been discovered through this route of analyzing familial susceptibility to particular types of cancer. And I've just listed some here: the APC gene, adenomatous polyposis coli gene, that's responsible for an inherited susceptibility to colon cancer; the RB gene, associated with retinoblastoma and characterized by Nutson; the TP53 gene, or P53, which is associated with many types of cancer, but particularly brain tumors in young kids, sarcomas in teenagers, and breast cancer in young women; the NF1 gene, associated with neurofibrosarcoma; two very important breast cancer susceptibility genes, BRCA1 and BRCA2; the INK4 gene, which encodes a kinase inhibitor that's associated with development of melanoma, brain and lung cancers as an inherited susceptibility; and then the one that I talked about at the beginning, MSH2, a mismatch repair gene associated with Lynch, with colon cancer. What do these tumor suppressor genes do? Well, they're mostly involved in inhibiting cell growth. So if we think about oncogenes, like the RAS gene or the RAF gene or the MET gene as promoters of cell growth, these tumor suppressors can be seen as inhibitors of cell growth. So they can block signaling pathways, they can inhibit the cell cycle, they can restrain the growth of cells. But they can also be very much involved in DNA repair, as we see with the MSH2 gene, and in maintaining genetic genome stability and chromosomal stability, and both P53 and APC gene are involved in this kind of maintenance of the diploid state. So another way that tumor suppressor genes were discovered and were found to be a way of probing their function was through the study of the DNA tumor viruses, and this is work that I was myself involved in and was a very hot topic for science in the 1970s, I guess. So this involved the use of small DNA viruses, so-called DNA tumor viruses, and there are many groups of these, but examples are SV40, adenovirus, human papillomavirus, which of course is very important for human development at risk of cervical cancer. So these are very small viruses. SV40 is only a 5.2 kilobase genome, And when the virus infects cells, there are a variety of different outcomes. So in human cells and monkey cells, the simian virus 40 can replicate its viral DNA and go on to produce new virions. In other cells, the cells are just not susceptible to the virus at all, and there's nothing happens. But in other cells, you can see that the virus is able to uncoat and starts to make viral proteins, and at some low frequency, the viral DNA can integrate into the host genome. And this happens particularly, for example, in the case of mouse cells or rat cells infected with SV40 virus. And then those cells that have stably integrated and continue to express the viral protein LT, large T and small t, those cells start to behave like transformed cells. So this was a very useful system in which one could take a virus, purified virus, and use it to transform cells from a normal state to a cancer state. But what made it exceptionally interesting at the time that I started working in this area was the discovery of temperature-sensitive mutants in the SV40 T antigen. So the SV40 T antigen protein is a large protein, multifunctional protein, is required for viral replication. So it was possible to select mutants that would only grow at 32 degrees, but not at 37 degrees centigrade. And when these mutant viruses were used to establish transformed mouse cells, the surprising finding was that the mouse cells were transformed at the permissive temperature, 32 degrees centigrade. But if those cells were shifted to 37 degrees centigrade, they started to grow back like normal cells. So the one actually had a reversible system. And remarkably, one could do this repeatedly. So you could shift between these two temperatures and see the cells changing in their behavior, their morphology, their metabolism, essentially a remarkable reversible shift in cell behavior. It's been exploited in a number of studies of stem cells, for example. And this meant a lot. It meant that not only was the T antigen responsible for the transformation event, for the altered behavior of the cell, but that the protein needed to be continually expressed to carry out this transformation function. So this idea of the sort of continued requirement for the expression of the oncogene distinguished it from a concept where the virus might have come in and essentially caused a mutation, what people used to call a hit and run type mechanism, but rather suggested that the expression of this protein was required to maintain the transformed state. So that led a number of individuals to suggest that this could be a very fertile area to study. What was the viral protein doing that could result in this transformation phenomena, and how could that effect be reversed? So this is a picture of myself and my colleague Keith Willison in 1979, and I was working then at Cold Spring Harbor, and it was at that point that I wrote up the paper describing the work that I'd done at the Imperial Cancer Research Fund with Lionel Crawford, where we first discovered the interactions between T antigen and a host protein called P53. So the paper was published in 1979. Makes me feel old to think about that. And what we discovered was that the viral T antigen actually interacted very closely with a very specific host protein in these SV40 transformed cells. And the way we did that was by using immunoprecipitation of labeled cell extracts. And we were able to do this because of technical improvements. Before this, immunoprecipitation proteins had been quite difficult, but we managed to use both protein A, which gave us a much cleaner immune precipitation mechanism than the ones that had been used earlier, which required anti-immunoglobulins. And then I had made a polyclonal antibody to the SV40 T antigen by immunizing with the purified protein, which I'd cut out from a polyacrylamide gel. So when we used this new antibody and this new immunoprecipitation method, we got very clean gels, and we could see a very clear band of T antigen being immunoprecipitated, but also a cellular protein as it emerged of 53,000 molecular weight. And we deduced from this that the T antigen was binding and interacting very specifically with a host protein. So subsequent to that, the tumor viruses have a common mechanism by which they function to transform cells, and that's to inactivate the function of two tumor suppressor proteins, the p53 protein and the RB protein. So the SV40 large T antigen does that by binding at its N-terminus to the RB protein and stopping the RB protein acting as a transcriptional inhibitor, and it binds to p53 and stops p53 functioning as a transcription factor, because when it binds to p53 it actually blocks its DNA binding activity. And all of these interactions were discovered by immunohistochemistry, by immunochemistry. Antibodies to large T immunoprecipitated these associated host proteins. And very excitingly, the adenovirus and HPV also target these same two proteins. So this just shows a crystal structure. This, of course, came out many years later. It's interesting to reflect at the time that we published those observations in 1979. They were met with a great deal of skepticism. People didn't believe that proteins could interact so tightly that they could be co-immunoprecipitated at the time. And we had a lot of criticism, people saying that all we picked up was a fragment of degraded T antigen. So though the evidence accumulated very clearly over the years that wasn't the case and that our hypothesis was correct, it was still a very nice day for me when the crystal structure finally came out, showing here's the hexameric T antigen co-crystallizing with p53 molecules that are binding to it. And from what we now know, these sites where the p53 interacts with T antigen are the same sites that p53 uses to interact with DNA in a sequence-specific manner. So by binding to T antigen, the p53 becomes unable to engage in sequence-specific DNA binding, explaining how the T antigen can inactivate this function of p53. So over the years, it emerged that all of the DNA tumor viruses really have these ability to interact with and inhibit the function of p53 and RB. So SV40 does it with one single protein T antigen, which binds both p53 and RB. Adenovirus has two proteins, the E1A protein, which binds RB, the E1B protein, which binds p53, and very importantly, HPV, which is a major cause of human cancer. The E7 protein binds RB, and E6 binds p53. And this is an example of convergent evolution, because if we look at the mechanisms by which these different proteins bind p53, they do it in different ways. So SV40 binds p53 through the DNA binding domain of p53, whereas the adenovirus recognizes another domain of p53. And the human papillomavirus E6 protein actually acts as a PROTAC molecule. It resurfaces an ubiquitin E3 ligase, so that now that E3 ligase can target p53. So the E6 protein acts as a kind of meat in the sandwich, and so cells in which HPV E6 is expressed, the p53 is degraded and disappears. So this has been a very important set of observations, and I think is still worthy of further study. Why do these viruses target these two particular tumor suppressor proteins so specifically? I mean, the current kind of argument is that, well, these viruses use host replication proteins, and so by for DNA replication. And secondly, that if they were to trigger p53, that might cause the cell to die by apoptosis. So by inhibiting p53, the virus wins because it keeps the cell alive longer while it makes more viral particles. So p53 is what I'll concentrate on for the rest of this lecture as an example of a tumor suppressor. And of course, one of the most important tumor suppressors, because the frequency of p53 mutation is the highest mutation frequency of any gene involved in human cancer. About half of all human cancers contain mutations in this particular gene. And you can see here, I'm sure you can't read this tiny script and I apologize for that, but essentially what it's showing is that mutations in p53 are the mutations that truncate the protein or a point missense mutations in the protein in blue occur in a huge variety of human cancers and at very high frequency. So in ovarian cancer, serous ovarian cancer, they're almost universal. In other common cancers like lung or colon cancer, about 50% or more of the cancers contain mutation in this particular gene. So what does p53 look like? What do we know about it? So it's a nuclear transcription factor that forms tetramers. It has a very unusual structure compared with many proteins because it's extremely unstable as a protein. It's very, very easily degraded. It's very, very easily unfolded. It has a DNA binding domain that's been crystallized, and then it has a transcription activation domain that is extremely unfolded, and an oligomerization domain allowing it to form tetramers, and then actually a C-terminal extension that's also unfolded. It's subject to an enormous number of post-translational modifications. So the protein is acetylated, ubiquitinated, netylated, methylated, phosphorylated, etc. And many of these modifications occur at different sites in the protein, particularly at the C-terminus and at the N-terminus. And central region of the protein is less highly modified, though acetylation of that region seems to be very important for its function. But what's interesting compared with other tumor suppressors like the RB gene is that instead of seeing p53 is mutated, but the gene is not lost, and that's because the types of mutations that occur are point missense mutations. So these are, in an individual cancer, you'll find an individual amino acid change, like this amino acid 175 can be changed from arginine to histidine. So we say R175H, arginine 175 changed to histidine, as a very common mutation, and many tumors will have that particular mutation, and these are called hotspots. However, as genome sequences has come to be realized on very large numbers of cancers, we realize that a huge number of different mutations are found in p53. In addition to the point missense mutations, there are lots of truncations, deletions, insertions, etc. So it's very, very highly mutated. And the other thing that's kind of driven a lot of interest is the expression of these mutant proteins, these point missense proteins in cancers, in cancer cells at high levels. And this is a very striking observation. And when we made antibodies to p53 that worked very well on paraffin sections, we were able to stain a very large number of tumors very quickly. So in the kind of the big breakthrough year for p53 was 1990, when the discovery by Bert Vogelstein and his colleagues of point mutation p53 was picked up by the field, and we were able to stain hundreds and hundreds of different types of tumors very quickly and see this overexpression of mutant p53 protein in very many different cancers. And very striking immunohistochemistry pictures are seen when you do this kind of staining. And the other thing that was noticed is that in the original studies of Vogelstein and others was loss of heterozygosity. So the way that sort of the role of p53 in human cancer was discovered was because Vogelstein discovered that there was loss of heterozygosity, chromosome 17p. He mapped the region where that loss of heterozygosity occurred to a relatively small region of 17p, and then sequenced the gene on the other allele, and found that there was essentially a loss of the chromosomal region. So typical human cancer will contain one mutant allele with a point mutation, and then a chromosomal event that's taken out the whole of the other allele. And sometimes the wild type, the mutant allele, is duplicated, so you'll see a duplicated chromosome, both of which contain the same mutation. This is just an example of p53 staining in breast cancer, I think in this case, and you see what the pathologist sees is this very striking, in this case we're using peroxidase and the DAB dye, so we see brown nuclei expressing high levels of p53, and the normal surrounding cells are not stained. So this is very characteristic. There's lots and lots and lots and lots of papers about p53 staining and the heterogeneity of the patterns of staining that are seen, but I don't have time to talk about that in detail today. But it certainly has been a very common observation and has driven a lot of interest. In some cases, it's very, very striking, the relationship between the morphological change in the cells and the occurrence of p53 staining. But if you look in normal tissues of a mouse or a human, there's very, 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 very low levels of p53 staining. So this type of high level of staining is very characteristic of tumor cells as opposed to normal cells. So a lot has been learned about p53, and I guess in the kind of golden era, if you like, was after 1990, and the next 5 or 6 years, sort of major discoveries were made about the function of this protein. So the first thing that was realized is that the protein is subject to ubiquitin-dependent degradation. And there are 2 proteins particularly involved in that, the MDM2 protein and the MDM4 protein. And both of these proteins, particularly MDM2, can be overexpressed or amplified in a number of cancers where it acts as a dominant transforming oncogene. And these 2 proteins normally function to degrade p53 through the ubiquitin pathway. So in normal cells, where the p53 promoter is on and the p53 mRNA is present and being translated, the levels of the protein can still be exceptionally low, almost undetectable, because this ubiquitin pathway is so efficient and so active. However, when the cell is stressed, this degradation pathway is blocked, and so a huge number of different stimuli can block this degradative pathway. There's one approach involves the activation of a small protein called ARF. The expression of this protein is very highly regulated, and when it's expressed, it acts as an inhibitor of MDM2, leading to the increase in levels of p53. And for example, the MYC gene acts very much through this pathway. So cells expressing high levels of MYC, their growth is inhibited because they MYC induces the expression of ARF, and ARF then activates p53. And so if you knock out ARF, a MYC transformed cell no longer activates p53, and those cells can grow very fast. The other major route, and these are all probably related, has been through the discovery of the link between DNA damage and p53. So DNA damage is detected by the ATM and ATR kinases, and subsequently downstream of those are CHEK1 and CHEK2 kinases. And those events all lead to inhibition of this MDM2, MDM4 degradation pathway, the phosphorylation of p53 and its activation. So high levels of p53 assemble on the chromosomal DNA, and often in the acetylated form, and then trigger downstream expression of a very large number of genes in the hundreds. And these genes are involved in induction of apoptosis, cell cycle arrest, senescence, DNA repair, alterations in metabolism, feedback regulation of the pathway is also very important. So one of the genes that p53 activates is in fact MDM2. So MDM2 is a p53 responsive gene, and that creates a kind of feedback pathway that sort of acts a bit like a thermostat on a radiator or air conditioning to control the levels of p53 in the cell. So in the same way as the mismatch repair gene and the retinoblastoma gene were discovered to be tumor suppressor genes through genetic susceptibility syndromes, in a slightly different way, Doctors Lee and Fraumini had described a very exciting cancer family syndrome in which children were subjected to very high incidence of early onset brain cancers and sarcomas, and remarkably early onset breast cancer in women. And these families were identified, and subsequently the syndrome is called Lee-Fraumini syndrome, and it's defined by a variety of clinical parameters in terms of the number of infected individuals, the type of tumors they get. But it was eventually determined in 1990 by David Malkin and colleagues that the gene that was responsible for this cancer family syndrome was in fact P53. So P53 mutations occur somatically in about half of human cancers, but when they're in the germline, they're responsible for this cancer family syndrome. And this is actually a conference I attended, which was rather well, moving, I think, and challenging. All these young people at the back here have Li-Fraumeni syndrome, and many of them have had serious operations to remove sarcomas, for example, in their legs or other places. But they were all attending this conference with great enthusiasm, and I think it was very challenging, actually, for us as scientists to realize that despite all the years of study of P53 and our understanding of its molecular mechanisms, we still are not in a position to do that much to help. Dr. Malkin, who was the guy who worked out that it was P53 that was responsible for this syndrome, and Arnie Levine and myself, who've both been kind of working on the P53 field for a long time. So Li-Fraumeni's syndrome occurs in about 1 in 10,000 births, and as I said, it's responsible for these cancer family occurrences. It's pretty amazing to realize that what's happening is just one base wrong. So these are point mutations, point missense mutations, and a number of issues have arisen recently around this area, which are common actually in the whole field of inherited cancer family syndromes, and that is what we call variants of unknown significance. So as more people are getting their DNA sequenced by 23andMe and others, people are getting reports that they have mutations in P53. Of course, that's a very disturbing piece of information to gather, but it's obvious that not every single mutation in P53 might be deleterious. So the question is which ones are and which ones aren't. And then the other thing that's caused a lot of trouble, I think, is this phenomenon that we've come to realize called clonal hematopoiesis, and that is in older people, individual blood cells can become very dominant. So you can get a sort of it's not really cancer, it's a very precancerous kind of overproliferation, but that can be misinterpreted as a germline gene because if people just sequence from the blood, there can be a mutant clone there that's not in the germline, but nevertheless has a high allele frequency. So again, as always, in the case of familial cancers, the analysis has to be very careful and thorough before information is given to the patient, because obviously huge decisions can be made about what to do next, and that's particularly, of course, in the case of the BRCA mutations, where women may undergo surgery as a result. And the other thing that's emerged is that things are more complicated than we thought, as they usually are in science. So though the P53 mutation is associated with Li-Fraumeni syndrome, there's one particular very large group of individuals who have a germline P53 mutation. And this is in Brazil, and there's about 90,000 descendants of a singular individual. And they all have this mutation R337H, arginine to histidine. It actually changes a single hydrogen bond in the oligomerization domain of P53, has a very subtle biological effect, and is associated with an increased risk in adrenal carcinoma in these kids of these individuals. But it's much less penetrant than some of the typical Li-Fraumeni alleles we see. So there's still an enormous amount to learn about these different mutations in P53 and the effect they have on individuals. So I think I'll get a bit philosophical now for my last 15 minutes. So, I mean, the field of P53 has become vast, and I just did this search. So this is 42 years of research. This page 24 of 2,220,000 hits. So it's very challenging for anybody in the field. This is a slide that I like that Karen Woosden produced, in which she showed the number of publications per year. And you can see after the discovery of P53, there are very few publications. There's a very small group of us worked on this mysterious protein that bound T antigen. And then in 1989, 1990, the whole field took off as it became clear that this was the most commonly mutated gene in human cancer. And in a very short period of time, people worked out about P53 being a transcription factor, regulation by MDM2, the story of Li-Fraumeni syndrome. They deduced many of the downstream genes of P53, and things developed into a very nice model of how it all worked. P53 as a transcription factor induced cell cycle arrest through a protein called P21, and it induced apoptosis through a protein called tumor. And we felt we understood P53 pretty well. But since then, I think it's fair to say things have become extremely complex. And with that number of papers, P53 has been reported to do almost everything you can imagine. And a huge number of papers are published every year describing yet more new findings about functions for P53. So calculate about 10 papers per day for the past 4 years. So I don't think any human being can have read all of these papers in any degree of detail. And so that raises all sorts of issues. It's a very kind of interesting field because many of the people who worked on the protein and the P53 system very early on are still in the field. And there's a kind of almost a club, I guess, of P53 scientists who meet every few years. And the next meeting is going to be in Rehovot in Israel in May of this year, COVID permitting, but I think it will. And that is both good and bad. So what am I going to talk about next? I mean, how do we make sense of this kind of level of chaos? And I think this is not unique to P53. It's a kind of feature of modern molecular cell biology that we've just got a huge storm of information. And how do we gather that information and make best use of it, make the most sense of it? So typically in a field like this, there's lots and lots of data. People are publishing many papers. People have lots of ideas of how it works. And a lot of those ideas are, I might say, dogma. That is, they become accepted, though when you look in detail at the basis of those ideas, it may not be as firm as it might be. There's challenges of integrating the different sorts of information. Newer methodologies evolve all the time. At one stage, everybody did microarrays, but nobody does those anymore. Now everybody does RNA-seq. It's a sort of big change in methodology. And I think, there's a problem of a lack of critical review of the data. And there's a human face of science. So this nice community of sort of cooperating scientists, which characterizes the P53 community, I do wonder sometimes whether it makes it harder to ask very difficult questions, which of course one should always do in science, because essentially we kind of know that we almost always are sort of wrong, or our knowledge is only partial. So I'm just going to sort of summarize that by just giving you a bit of dogma first, and then discuss some of the issues that have arisen really by looking carefully at new databases. And these relate to the role of P53 in the response to DNA damage, and whether this is really how P53 works to suppress tumor formation, and how important is the transcriptional function of P53. P53. This should be taken with a big pinch of salt. So dogma, what are the consequences of missense mutations in P53? So the first consequence is loss of wild-type P53 activity. And I think this is not controversial, and as we'll see in a minute. The second concept that's arisen is the concept of dominant negative inhibition. That is, if you are making a mutant P53 protein, that it can inhibit the function of the wild-type protein. And there are many, many papers reporting this finding of dominant negative inhibition. And the final concept is the idea of gain of function. So this is where the idea is that the mutant P53 protein has a new function, a new activity. You're expressing large amounts of the mutant protein, and this mutant protein is actually acting, if you like SU40 tianjin or RAS as an oncogene. And these kind of ideas are very, very popular, and there'd be many, many papers in Israel describing all of these concepts, and particularly this area of the gain of function of mutant p53. And if you look at why people believe in this, I mean, there are lots of very good bits of data that would support their idea. And so one of the most important of these two papers that were published in 2004 functions. If you look at a wild type mouse with two wild type p53 alleles, it has a very low tumor incidence. Larry Donohoe made p53 system that these animals are viable and fertile, but they are prone to developing cancers, and they develop particularly lymphomas and sarcomas. So the interesting question is then what happens if you have a point mutant like the R175H mutant allele over a null background? So now we're just asking the question between a mouse that has no p53 at all and a mouse that's making just a mutant protein. So in this case, we're not looking at the dominant negative phenotype, we're looking at the gain of function phenotype. And what was reported was that these animals differed from the null mice by having, in addition to lymphomas and sarcomas, carcinomas that were more invasive and metastatic. So this, at face value, is a compelling piece of evidence that the mutant protein is sort of altering the types of cancer that the mice get, though it doesn't actually, even in the original publication, alter the number of animals getting tumors, or indeed the rate at which they die, but it does alter the type of cancer they get. And I mean, it's very tricky to say that this observation very recently, another group has published that they couldn't see the same result, and so it's very hard to know what to make of that, but that's what's get at these questions. Well, we can start to look at new data sets, and where the whole gamut of new methods has been applied to a very large number of human tumors, and these methods include obviously whole genome sequencing, but also ChIP-seq and mass spec analysis and use of CRISPR libraries and other new technologies. And so one of these papers, which was published a few years ago now, not so long ago, in 2019 by Larry Donohoe, was to sort of take all of the published databases and analyze them carefully, and try and integrate the information that had been gathered by those large-scale studies. So this paper looked at 10,225 cancers, and then looked at the whole exome sequencing, and was able to then split them up into those that were p53 mutant and those were wild type for p53, and then look at things like DNA copy number, RNA expression, mRNA expression, protein expression, and analyze what was happening in terms of things like a prognostic signature, where you could say whether p53 was functional or non-functional, not just looking at mutations in the gene itself, but looking at downstream consequences, look at the stability of the genome, look at the question of whether p53 was biallelically inactivated or not. So 5 data platforms in 32 cancers. So the first finding is that essentially you always lose the wild type allele. So it's loss of the wild type allele is very common, and by itself that already tells you that the dominant negative phenotype of mutant p53 cannot be completely penetrant. Otherwise there should be no selection to lose the wild type allele. If you make a mutant protein and it binds to and inactivates the wild type protein completely, then there should be no selection. And then the finding that mutation in p53 affects globally RNA and protein expression and has a very big effect on genomic stability. And finally, that using the mutant p53 RNA expression signature rather than simply using whether p53 is a mutant or not, gave helpful prognostic predictions in 11 different cancer types. So this database allows you to look at lots of very interesting things in a kind of very comprehensive way. In human cancer in p53, and this is a frequency of observed mutations and variants that are never altered. And what you can see is this amazing high frequency of mutations almost throughout the whole of the p53 protein, but particularly focused in the DNA binding domain. But there are plenty of variants associated with the oligomerization domain and the N-terminus as well. And this kind of pattern is very striking indeed, and it suggests that p53 is very, very easily mutated. So if you imagine a protein that's folding structure is not very firm, if you like, it's a protein that's almost molten, almost unfolded at body temperature, then you can see how many different mutations could affect it. It's very different to, for example, a multi-domain protein, which might have regions that can be mutated while retaining other activities. So p53 is very, very easily mutated. And what's emerging is that some of these mutants, rather than gaining function, they actually retain some residual wild type activity. So in fact, the mutants that are most common are the ones that have completely lost wild type activity, and the rarer ones are the ones that have retained a little bit of wild type activity. So really, the TCGA data challenges two dogmas. It challenges the dominant negative dogma, because though the dominant negative effect is very easily seen in cell biology, clearly it's not fully penetrant. You could argue that it has some effect, and therefore it sort of acts as a promoter of instability, helping to lose the other allele. But it's not very clear that that's the case. And I think p53 is a tetramer and functions as a dimer of dimers, and the dimers assemble on the polysome. The mixed tetramers are inactive in DNA binding and transcription assays, but how often do these mixed tetramers actually form? So what we can deduce is, why do we always have to lose the wild type allele? It's because we can't afford to retain any wild type function. And so there's a strong selection against any residual wild type p53 activity. And if you think about a dominant negative model, you're going to have, if you have equal amounts of the two proteins, you will of course, have these equilibrium events. And so these will be the inactive forms. If blue is mutant, all of these will be inactive, but you will still make some active p53, and that may be more than sufficient to carry out the tumor suppressor function. So this is the dominant negative effect is not very convincing in these databases, and it's particularly clear that you don't see a particular difference in the outcome between truncating and missense mutations in terms of, for example, % survival. So here is wild type genotype, and here's a truncating, and here's a missense. So only the missense can show dominant negative. The truncating can't, because it doesn't have the oligomerization domain, which is at the C-terminus of the protein, and yet there's no really clear difference. So if the dominant negative model was very important, you'd expect to see the missense mutations having a worse survival than the truncating mutations, but you don't see it. And you can see the same problem when you look at the germline inheritance as well. So essentially, in the Li-Fraumeni data, we do find in Li-Fraumeni families with truncating mutations as well as point missense mutations, and yet we don't see any big difference in the behavior of those individuals in terms of their onset of cancer or the type of cancers they get. So if we're brutal about this, we could say that from the TCGA data, though there's evidence, of course, of loss of wild type p53 activity, that the importance of dominant negative inhibition of wild-type p53 or acquisition of transforming function, gain of function, is not really clearly supported by those data sets. And then, of course, as you know, WEHI itself has been the source of a lot of trouble in the beautiful models of p53 we used to have, where p53 induced p21 and puma, and that results in cell cycle arrest and apoptosis. And that's certainly important for what we would consider to be the damaging radiation response. And you can show here by looking at the comparison between p53 wild-type mice and p53 null mice, and looking in green at tunnel staining, that you can see that in the wild-type mice, you get radiation-induced apoptosis in a number of different tissues, thymus, spleen, small intestine, bone marrow, and that's really absent from the p53 null mice. So this kind of acute damage response resulting in apoptotic death of these hematopoietic cells is very important for a response to chemotherapy and radiation therapy, but it's probably not important for tumor suppression. Andreas and his team's work, where they made mice that lacked p21, puma, and noxa, these animals essentially had lost that acute radiation response, but they are not tumor susceptible, whereas the p53 knockout mice are still highly tumor susceptible. So this suggests that there are other things that are happening that are important for tumor suppression. And though we have this very nice model of the acute DNA response, it doesn't seem to be what's really causing tumor suppression. And that's supported by other work by Gerard Evan using an inducible system. So we have to face up to the fact that this is probably not the mechanism of tumor suppression. And the other thing that sort of confounds the model is mice made by Laura Tardy, in which 2 point mutations have been made that make p53 a very, very weak transcription factor. And yet these animals are still tumor-free. So there is a little bit of residual activity of p3, but it's very small compared with that seen with the wild-type animals. So what's going on? I think we don't know. I've just published a paper myself in PNAS saying that p53 is very involved in damage recognition and DNA repair. And there are lots of other hints of p53 doing other things, certainly maintaining the diploid state is very important. This is an experiment by Gerard Evan with an inducible p53. So when the p53 is on, you see very nice G1, G2, but when it's off, you see lots and lots of aneuploid cells arriving, and that's similarly a frequent event in p53 knockout cells. And this is a very common feature of fibroblasts examined from Lee Fraumini families. So this genetic stability at the chromosomal level is important, and recently a new pathway has been discovered in which p53 is very involved and regulated by BP1 and USP28 in the regulation of controlling centrosome loss and the length of mitosis. And there's also more recent papers showing p53 being recognized and degraded following its methylation by a completely new pathway. Of course, it's interesting to look back, and this is a very old paper from Arnie Levine in which he says that p53's prevention of mammalian DNA, followed by release from mitotic spindle type point, requires p53 to protein, but not its transcriptional activity. So Jennifer Piettenpohl has sort of looked at this issue as well, and just has really come to the conclusion that the acquisition of aneuploid mimics what many others have associated with p53 as a gain of function. And again, she shows that, you know, clinically p53 loss of function and aneuploidy show the same importance. So if you look at the fraction of genome altered, you get similar numbers from both truncating and missense mutations, and it really does suggest that most of the characteristics ascribed to the gain of function can actually be explained simply by the fact that you've got aneuploid cells. So I'm going to finish there and say, you know, how do fields get stuck? Is dogma useful? tilt wouldn't. And does the publish or perish model inhibit our challenge? Do personal relationships inhibit challenge? It's quite uncomfortable to tell your best friend from many years that you think their ideas are completely wrong. Who's reading the 90,000 papers and who remembers what they said? And should we have some sort of consensus review about what we think is happening in the field every few years? And is this the most efficient way to do science? I've become very interested in the Cancer Grand Challenge approach, where you bring teams of scientists together with different approaches and different technologies to try and answer a particular problem. And maybe we should all spend more time, like Alfred Nutson did, thinking about our problem rather than making new experiments. So maybe we need more coffee and less pipettes. Now, at that, I'll say thank you and look forward to hearing your questions.
Thank you so much for that amazing talk. Yeah, so for this, anyone here, if you want to ask a question, just raise your hand using the raise your hand feature, or type a question into the chat box. So, and I know there's students that have to ask questions, so we'll see. Oh, Gemma. Hi, Gemma. Would you like to go first?
I will. I'll start the questions off. So we're, as you know, we're probably very interested in...
You can stop sharing your screen if you would like.
Oh, yeah. That's a good idea. So we're interested in the gain of function, and I guess, you know, looking at the evidence that maybe there's not this strong gain of function, so maybe mutant p53 itself is not a therapeutic target. So what does that leave us with? You know, where do you think the field should be heading in terms of looking for more efficient therapies for people who have mutant p53 cancers?
Yeah, I mean, of course, it is very attractive to think of the mutant proteins as a target, but if we accept, I think we probably should, that it's not so clear, then, you know, you're stuck really with I think with thinking about aneuploidy as the key characteristic, or, you know, something to do with chromosome loss. And I guess the model is obviously BRCA and the PARP inhibitors, and maybe we should be looking for that kind of synergy, that kind of susceptibility created by the aneuploid state. And I think, you know, that's something that needs to be thought about. I mean, it's very difficult. I think we've tried very, very hard with the MDM2 inhibitors, and I mean, they've made some progress, but we do know that the escape from MDM2 inhibitors will be very straightforward, which will be p53 mutations. So, you know, I mean, it's not to say that they won't be useful, but they're not. It's not absolutely clear. I mean, I think we just have to start, you know, thinking differently. I guess immune effects of the loss of p53 are something that are very interesting to think about. If it does create some sort of capability for the cells to be recognized by the immune system, that would be very powerful to look at. So I think those are kind of directions we can think about. But yeah, I mean, it's a continuous frustration. I suppose, Arnie Levine would say, and others would say, well, maybe we can get the mutant protein to regain function. But I think that's also being worked out very hard, and it's proving extraordinarily difficult. So I don't know. I'm very frustrated, I guess, like everybody. It's sort of like you've been given this huge chunk of knowledge. Here's this common feature of human cancer, and then, oh, actually, it's not helping.
Thank you. It's helpful.
Okay, so we have a question from Erin, and she asks, is there a push to identify tumor suppressor gene mutations in the general public prior to cancer development? And can mutations that will lead to cancer be recognized as biomarkers?
Yeah, I mean, I think that's a very good question. I mean, I think people are beginning to look for inherited susceptibility more broadly. So you see it particularly in breast cancer, where there are quite a few different alleles now. We know that, you know, BRCA1, BRCA2, p53 only account for about half of the familial risk, and the other half is probably coming from a combination of other mutant alleles of different genes, which sort of quite minor in their effect. But if you get, a bad hand, you get 3 of them, then you've got a problem. So yeah, people are beginning definitely to do that, to look at a wider variety of variant alleles to see if you can pick up a at-risk group that might then be more frequently screened. And I think that's sort of the direction that people are thinking about. And I think it makes a lot of sense. I mean, we are clearly going to get to the point where everybody's genome sequenced, and sooner rather than later. And I think looking then for modifier genes, if you like, is going to be very important. It's already in the Brazilian cohort, there's some evidence for modifier genes. So some of the families have a much higher risk than others of developing a more Li-Fraumeni-like syndrome, whereas other families seem to have relatively little risk, or they have a very specific risk for adrenal cortical cancer. So clearly, I think that is a good way to go, because I mean, Li-Fraumeni is so interesting in the sense of a model for us to study of at-risk individuals, and can we show prevention methods, screening methods working in those individuals. And David Malkin and his colleagues in Canada have done an amazing job, and they had, I think, 2 papers in the New England Journal 10 years apart. They have a cohort of kids where they've screened them very carefully, and they definitely do show extension of lifespan by doing that. But it's not without cost, because the screening itself is very invasive, and often the treatment is surgical. And they're kind of disturbing things. So they can look back through all the radiographs and say, okay, this tumor arose. When did we first see it? And you see it in one scan as just a tiny, tiny dot, and then by the next scan, it's already quite a big tumor. So, but it could have been the tiny dot for 3 previous scans. So it can kind of sit there doing nothing, and then boom. So I think it's there are great challenges, but they're great models, great systems to work out those challenges.
Okay, so Jeffrey asks, have P53 mutations been found which reduce cancer rates?
No, I mean, but that's a very interesting idea. I mean, the nearest we've come to that is the experiments where they introduced an additional allele of P53. So essentially, there are mice which have an additional copy of P53, and if it's correctly regulated, that can reduce cancer incidence. I mean, you could argue that it is the case already, and that we just haven't looked carefully enough. So I mean, Arnie Levine showed that there's a SNP in the MDM2 gene that results in higher or lower level expression, and therefore higher or lower levels of P53, and that SNP is associated with a higher incidence, higher risk of cancer. So essentially, the level of P53 can be very important. So it's not unreasonable to do a population-based study, if you like, to ask that question. I don't think anybody's done it yet, really. I mean, Arnie's study with MDM2 was the closest anybody's got to doing that. But yeah, I'm trying to remember the name. There's a group in Oxford that have also been looking at that carefully to see if they can see, individual differences in the P53 response that are meaningful.
Okay, so Sophie asks, do mutations within the oligo and transactivation domains of P53 cause similar amounts of dysregulation as mutations found in the binding domain of P53?
Yeah, I think the most data we have is the oligomerization domain, where they do cause disruption, but it's not as great. Transcription activation domain has been rather difficult to get single point mutations that have much effect. So that most of the mutants are double mutations, which are very rare, not going to occur at all frequently in the population. But again, there's a lot of work to be done. I mean, part of the problem is as we've got more and more sequence data, we get these variants, but we don't know whether they're critical. I mean, if we get a family, of course, where we can see familial inheritance, then it's very clear. But I mean, that can be difficult. So I did see a recent case report where they had a mutation in, it's a single amino acid change in right at sort of border of the transcription activation domain, the DNA binding domain, and give a very clear Li-Fraumeni syndrome with very good genetic inheritance. So it's I mean, every family is interesting, actually. I was a bit surprised when I went to Li-Fraumeni conference how few of them had what I would consider normal mutations. They all had weird mutations. So I think, you know, as always in the field, people take the simple example first, and then it gets more complicated. So, but yeah, I think that there's a lot more data coming out, but each of those alleles has to be studied carefully.
So Tamer asks, how similar is E6 binding to P53 to MDM2?
Completely different. Let's say there is fortunately a crystal structure for E6, and it binds to the DNA binding domain, and it's a very subtle and interesting interaction. But yeah, it basically resurfaces an E3 ligase, a HECT E3 ligase, and this biology is PROTAC actually, and it's very good. So that's exactly what it does. Yeah.
Okay, and Michaela asks, P53 is frequently mutated along with other genes. What are your thoughts on P53 being a passenger versus driver mutation?
Yeah, that's a good question too. I mean, I think there's a lot of evidence from sort of truncal analysis, you know, the sort of tracer X, that P53 is retained. It's not, the mutation is always present in all the progeny, if you like, of the cancer clone. And clearly in ovarian cancer, you know, it's always present. I mean, you always get P53 and then you get RB as well. But, you know, could it be, in some cases, you know, in colon cancer, it's reported to be a later mutational event. I think all of that is still a bit up in the air. I mean, our whole understanding of what's going on with mutation in cancer is changing as we've realized that so many of cells in our body already got mutations in P53 and other genes, and it's more variations in promotion that seem to be increasingly important. So I think it's, yeah, I mean, it's a good question. I don't think it's completely resolved. I mean, clearly in some cases, it's not, I mean, to call it a driver, I don't see it in the same way as one might see an oncogene. It's not necessarily driving the mutation. It's basically allowing things to happen. You know, it's like losing your brakes rather. And I guess one of the problems in the field is that, you know, there's sort of backup mechanisms. So, you know, it's like a dual circuit brakes, or you know, on an aircraft you have 3 different hydraulic systems for the brakes. So you lose one, you lose 2, it still looks like it's okay, but there's a greater risk of the third event. So I think it's quite it's more like that really. I mean, yeah.
Thank you. So does anyone have any last questions? You can raise your hand. If not, I'll ask one.
Okay.
So what do you have any thoughts of like why particular mutations are more common in certain types of cancers? Like you said, that Li-Fraumeni family, although low penetrance, has like a similar phenotype. What are your thoughts on this?
Yeah, I mean, I think it's the only case where it's particularly well, no, that's not fair. One case where it's particularly interesting is this adrenal cortical cancer, where it does seem to select for a particular subset of mutations. And in fact, now that, you know, if you look in kids with adrenal cortical cancer, they nearly always have P53 mutation of this particular subset. I mean, one theory, which I don't know if it's true, is that particular mutation, the protein is kind of is so nearly normal that it's only in certain cells that you see the abnormality. And it was, somebody suggested to me that it might be to do with the pH of the cells, you know, that they, for some reason, they had a slightly different pH and so forth. This hydrogen bond was therefore more fragile in that particular environment. I mean, it's a fascinating issue in terms of, you know, why you get these effects. I mean, it's really dominant, concept in the whole field of tumor suppressors. I mean, why does RB is associated with retinoblastoma, and individuals inherit that germline risk? But we know in somatic mutations, RB mutations are very frequent in lung cancer, but we don't see a huge instance of lung cancer in individuals that we know are carriers of the RB allele. So there's a lot we don't understand in terms of the tissue specificity of why a suppressor is, you know, particularly effective in one tissue versus another.
Okay, thank you. So I know it's pretty late for you, but we've got one more hand up. Are you okay to answer one more question?
Yeah, one more, yeah.
Okay, so Anna, can you ask your question, please?
Can you hear me?
Yeah.
Yeah, can you? Thank you so much for answering my last question. I just had this philosophical last-minute question. Probably you've answered it already, but I was just really wondering, so all these tumor suppressors, and especially P53, that seem to have such a major role, or maybe primary role in suppression of a progression of a wrong, like, I don't know, growth of cells or something. Why are they so kind of, yet they seem so fragile, they seem not protected well, seem to have been predisposed to so many PTMs, or like, you know, so many genes regulate them. So why, I was just thinking our biology would be the way that one of the most important genes would be so like open to all these modifications and stresses, and why they as you said, like be more protected.
Yeah, terrible design, isn't it? No, I mean, it's a very good question. I mean, why are we in the, you know, it's very bizarre to me that you can have a single base change and give this big problem when in a system that's so, you know, so otherwise so redundant. I think we can't, you know, it's very difficult. Evolution just happens and things work out, and we're all supposed to die when we're about 30 anyway. So we're in we're a very strange situation of living longer than the sort of evolution selected us to live. But yeah, I don't know. I mean, I have no idea. I find it very curious. And I mean, obviously that why, you know, it's a bit the same question, you know, why sarcomas, why retinoblastoma, you know, what is it that's creating these particular needs or specificities? But yeah, it's terrible design.
Yeah, it kind of doesn't make sense if we want to survive.
No, it doesn't.
Thank you so much, David. That was very
pleasure.
Okay, so thank you once more, David, for coming along and giving us this fabulous seminar. To all the students, we will see you next week at the next one, and yeah, have a good week.
Okay. Night, everybody.
Great. Thanks, David. That was fantastic.
Well, good morning.
We'll let you go.
See you soon. Yeah. Okay. Bye.
We'll let you go and get some sleep. Thanks very much.
Thank you.
Thank you.
Bye.
Bye.